Reference intervals. Test results are generally expressed in number with units, reflecting the amount of analyte in a given volume of fluid concentration. The results of patient tests are compared to a reference interval, a range that has been documented to reflect the result expected for healthy people. There are several ways to define a reference interval. Some reference intervals are based on consensus values that reflect medical decision levels. These values are agreed upon by healthy care professionals as good indicator for medical decision making. Some reference intervals, especially for tests where there is no medical consensus value, are based on statistical analysis of results of the test for healthy population. Consensus reference intervals, when test results can be correlated with medical decision level. Reference intervals are determined by consensus of healthcare professionals. The values are based on the result of clinical research and the clinical experience. For example, the American Diabetes Association has used the results from many clinical research trials to develop consensus value for blood glucose and hemoglobin A1C. The American Heart Association and the expert panel from the National Cholesterol Education Program have evaluated the role of lipid as a risk factor for heart disease, and based on many research studies of heart disease have identified desirable range of cholesterol, triglycerides, HDL, LDL. Example of consensus reference intervals: medical decision level for clinical chemistry test, glucose fasting 100 mg per deciliter, cholesterol 200 mg per deciliter, American Diabetes Association, American Heart Association, triglycerides 150 mg per deciliter, 1.5 millimole per liter, American Heart Association and National Cholesterol Education Program. Prostate specific antigen PSA 4 nanogram per mil, 4 microgram per liter, American Cancer Society. Hemoglobin A1C normal less than 5.5%, less than 39 millimole per mole, American Diabetes Association and the International Federation of Clinical Chemistry. A statistical reference interval: when a test doesn't have a clear association with a single disease or condition, or when there is insufficient medical evidence to define specific reference interval, a statistical approach is taken. The reference interval is typically based on statistical distribution of values obtained when the test is done on hundreds of healthy people. The range of results identify the values that are most often seen in healthy population for a typical bell-shaped distribution. As shown for calcium, approximately 66% of all results are within one standard deviation of the mean. Approximately 95% of all values are within two standard deviations for the mean, and approximately 99% of all values are within 3 standard deviations of the mean. The reference interval is generally chosen to capture central 95% of healthy people and is said to be the range from -2 SD to positive SD, referred to as 2 SD range. Sometimes it's said to capture the central 99% of healthy people and is said to be the range from -3 SD to positive 3D, referred to as 3D range. For the example given for calcium, a 2D reference interval would be 8.6 to 10.2 mg per deciliter, whereas a 3 SD reference interval would be from 8 to 0.3 to 10.6 milligram per deciliter. In first case, 5% or 5 out of every 100 healthy people would be expected to have a test result outside of the reference interval, either high or low. In the second case, only 1% or 1 out of every 100 healthy people would be expected to have a result outside the reference interval. Most chemistry tests use 2SD reference interval, while some tests like troponin use 3SD reference interval 90 percentile to separate healthy patients from those with a disease. Many analysts, however, don't demonstrate the normal distribution in healthy population value and may be skewed toward one side of the mean or have extreme high or low tails of value that distort the bell-shaped curve. In this instance, the reference interval can be estimated using non-parametric statistics that make no assumptions regarding shape of the curve. In non-parametric analysis, result values are ranked from low to high, and 2.5% of value are excluded from each end of the defined central 95% of value. A 2SD reference interval is a value that encompasses the central 95% or mean, positive, negative 2SD of distribution of result of healthy population, regardless of where the results are bell-shaped or non-irregular distribution. Similarly, a 3SD reference interval would comprise 99% distribution of result from healthy population. If the range of values seen in healthy people tend to be near zero and there is no medical concern about the low value, the reference interval is sometimes expressed as 0 to a number that represents the upper limit of the 95 or 99 percentile of the healthy population. May also simply be expressed as less than that number. Expected value may vary among different healthy populations, and different reference intervals are often reported for those populations. Most common differences are those based on gender, age, ethnicity. Specific population range are statistical range determined for each population based on the choosing partition factor.